Today we will be talking about the design principles used by nature. We will see the remarkable effect of this design. What we are going to discuss is that why rate of electron transfer is so fast. We will take an example of blue copper protein and we will see soon that how design principle is so important. Now, why rate of electron transfer is so fast in blue copper protein? Blue copper protein, as you all know, found in a remarkable varieties of organisms ranging from bacteria to human, and rate of this electron transfer is very high, 10 to the power 4 to 10 to the power 6 mole inverse second inverse. What exactly happening in blue copper protein? Basically, it is interconversion between copper 2 and copper 1. Copper 2 takes an electron, gives rise to copper 1, and copper 1 releases an electron, converts to copper 2. Now, for 4 coordination, what you can see that copper 2 prefers to stabilize in a square planar geometry. In contrast, copper 1 stabilize in a tetrahedral geometry. Now, this being so that a huge reorganization energy is required for such switching square planar to tetrahedral and tetrahedral to square planar, and how does electron transfer occur so fast? Now, before we discuss about blue copper proteins in details, let us look at the characteristics of classical copper 2 centers in proteins. There are 3 types of classical copper centers. The type 1, which is called blue copper center, and as you can see here that the function is reversible electron transfer. So, copper 2 takes 1 electron, becomes copper 1, and copper 1 becomes copper 2. So, the structure of these blue copper centers in the resting state is shown over here. You can see that copper center is ligated with 2 histidine, 1 cysteine sulfur and 1 med sulfur thioether sulfur, which is giving a weak copper sulfur bond. Now, in the oxidized state, they have some remarkable features, like they produce an intense blue absorbance at nearly 600 nanometer, and EPR spectra is uncommonly different from other, produce small hyperfine splitting in G parallel region. In contrast, copper 2, which is known as non-blue copper center, its function is that dioxin activation from copper 1 sites in cooperation with organic enzymes. So, if you look at this active site structure, is basically this copper center is ligated with 3 histidine and 1 weak water ligand is coordinated at the 4th position. And the characteristics features of this oxidized species is typically it produce weak absorbance and less than 1000 mole inverse centimeter inverse epsilon value, and also basically it produce normal copper 2 EPR. Now, there is another type called type 3 copper dimers. This kind of molecule you see when you see that oxygen uptakes from the copper 1 state, and this is a dimeric structure, and the in the oxidized form there is a strong absorption in the near UV region nearly 300 nanometer, and because of the strong antiferromagnetic coupling between 2 copper centers, these copper 2 are EPR inactive. Now, let us come to their EPR spectra. As you can see that that copper 2 hexaqueous species gives rise to an EPR signal as shown. As you know that copper is I value is 3/2. So, there will be 4 lines, and because of electron nuclear spin hyperfine interaction donated by the parameter A, anisotropy of the hyperfine interactions is manifested by components parallel and perpendicular to the molecular axis, namely A parallel and A perpendicular. As you can see that this A parallel, the separation between these two signals, there are 4, 1, 2, 3, 4 signal at the parallel, and there are perpendicular regions are here, and this separation is A parallel. And if you look at that type 1 and type 2, you see that in type 1 this A parallel is very small, whereas in type 2 you see that A parallel is quite large, that is normal copper 2 center, but type 1, which is actually blue copper proteins, produces this type 1. You see that very small A parallel value, and this is quite interesting, and we will talk about that. Now, coming back to blue copper proteins, is a large number of proteins are actually comes within this family of blue copper protein. Some of them are azurin, plastocyanin, amicyanin, stellocyanin, and the active site structure of this blue copper protein is more or less like this. As I have said earlier, this copper center is strongly coordinated with 2 histidine ligand and 1 cysteine sulfur, 1 med sulfur, and there is a additional X is also found to be present, and this X is sometimes O donor from carbonyl group at a distance of 2.8 to 3.2 angstrom. Now, 2 representative blue copper proteins are shown here. One is popular plastocyanin, 2 histidine are strongly coordinated, and then cysteine sulfur also very strongly bound with the copper, but this med sulfur is weakly coordinated. In azurin, in contrast, you see that it is very similar with the popular plastocyanin. However, there is an X which is coordinated to carbonyl oxygen, and med sulfur is also weakly coordinated. So, these two are weakly coordinated to copper, and these 3 bonds are strongly ligated, and we will see soon their spectral features. Now, crystal structure of azurin is shown over here. You see there is a huge protein chains which are wrapping around this copper center, and as you can see that copper center strongly ligated with 2 histidine nitrogen and 1 cysteine sulfur, and these other 2 are just very weak bond. Once we remove this protein, you can see very clearly this dotted line are very weak bond in the 4th and 5th position. The crystal structure of popular plastocyanin is shown here. Here again this copper center is buried within this protein chains, and you can, if you remove this protein, then you see this copper centers clearly, which is ligated with 2 histidine and 1 cysteine sulfur and 1 med sulfur thioether, and this is of course this is a long bond, copper sulfur bond is very long. This is the structure of popular plastocyanin in the resting state. Now, some of these bond distances are shown here, as I have just said that this copper nitrogen, histidine nitrogen are strongly bonded 1.19, 1 2.06 over here. Copper cysteine sulfur is also strongly ligated 2.07. Interestingly, copper med sulfur bond is very weak, 2.82 angstrom in popular plastocyanin. In contrast, azurine also, this copper nitrogen, this histidine to histidine and the cysteine sulfur distances are also very short, that means they are strongly bonded. And as I have said earlier, that fourth and fifth ligand like copper met sulfur distance is 3.15, very weak distance, and copper oxygen is also 2.97, again very weak, which is actually often in the X structure of azurine. Now, some of these spectral features are shown over here in popular plastocyanin. X structure is shown here, and this is what is the absorption. You can see that the huge difference between blue copper and normal copper, and EPR also shown here has been compared with the blue copper protein and normal copper, and there is a huge difference. Now, I will discuss one after another, like electronic spectra of copper to center in popular plastocyanin. As you can see, this the spectra is compared between normal copper and blue copper protein, and the absorption bands actually responsible color of simple cupric complex, however, typically have extinction coefficient of only 5 to 10 mole inverse centimeter inverse, which is clearly reflected. As you can see, they are all low intense bands. In contrast, what we see in blue copper protein, that there is an highly intense band around 600 nanometer with an extinction coefficient of more than 5000 mole inverse centimeter inverse. So, huge spectral change between normal copper and blue copper. If you look at the EPR spectra of this copper to center, you see that in case of normal copper, this A parallel is a very large 164 into 10 to the power minus 4 centimeter inverse. In contrast, in blue copper protein, it is only 63 into 10 to the power minus 4 centimeter. So, EPR spectra of blue copper proteins exhibits unusual features, including high G values and low A values, suggesting that unpaired electron is more delocalized than in other copper 2 centers. Now, let us little bit talk about rate of electron transfer. Now, as you all know that in Marcus theory, this KET, this is the expression, and you see that KET is that the electron transfer rate, which is related to HAB, which is called electronic coupling factor between the reactants. Also, delta is depends on delta Z0, which is the Gibbs free energy change upon electron transfer, and one very important factor which is called lambda reorganization energy. Here you see the reorganization energy, which is basically described as the energetic cost related to the conformational change between reduced and oxidized species. So, this is the factor which is actually changes from one molecule to another molecule. Now, we will talk about the geometry preference of 4 coordinate copper centers. Like copper 2 and copper 1, they are interconverting between them. Now, copper 2 actually is a D9 system and copper 1 is a D10 system. Now, D9, as you know that it prefers to stabilize a square planar geometry, whereas D10 prefers as a tetrahedral geometry, because then the ligand-ligand repulsion would be minimized. Now, changing from square planar to tetrahedral and tetrahedral to square planar is a drastic geometrical change. Okay. So, there is a drastically different preference in the coordination geometry, and if this is so, then how does electron transfer occur so rapidly in blue copper proteins? So, blue copper protein structure is shown over here. As you can see that this angle cysteine sulfur, copper and mid sulfur, this angle is 109.7 degree. Basically, this is a highly distorted tetrahedral structure with a very long copper thioether bond of 2.90 angstrom. This is high energy molecule, high energy geometry, and it has been said that this is in enstatic state, is high energy state. This unique geometry of metal center accounts for minimum reorganization energy, and that is the reason why rate of electron transfer is so fast. Now, let us talk about enstatic state or rack state. Basically, this copper 2 and copper 1, they require normally a huge reorganization energy in geometry while transforming between each other. However, in the blue copper protein, we have seen that the geometry is distorted tetrahedral structure both in copper 2 and copper 1 state. Now, this is certainly a high energy state which is called enstatic state, and enstatic state has been coined by Valley and Williams in 1968, which basically they define as a closure to a transition state that to a conventional stable molecule. You can see that is an hypothetical diagram is shown over here. This molecule is close to the transition state. So, it can go to this direction or the other direction very easily, because reorganization energy is very less. It is neither square planar, not a pure tetrahedral. So, it is actually a distorted tetrahedral structure, and both copper 1 and copper 2 have the same geometry. Now, let us look at that how in enstatic state the reorganization energy is reduced. As discussed, that copper 2 and copper 1 has completely different geometry. However, if you look at the extra structure of both oxidized and reduced state of blue copper protein and overlap with one another. So, what you see? You see that there is a very small change between oxidized and reduced state. So, the geometry is so rigid that such small change in geometry upon redox in plastocyanin has been attributed to restriction of the active site by the protein in an enstatic or rack state. I am showing you once again the protein structure of popular plastocyanin. As you can see that all these coordination sites are actually very rigid, and protein actually creates those rigidity, and they stabilize this distorted tetrahedral structure both in oxidized and reduced state. Now, once you remove this protein, you can see the structure. However, the copper ion can be often be removed from blue copper proteins by the treatment with cyanide. The apoprotein obtained in this manner was crystallized and found to be isomorphous to the metal containing forms, indicating that the structure undergo very little change upon metal removal. So, this is clearly reflected in the reorganization energy. As you can see in blue copper protein, the reorganization energy is very low. It is between 0.6 to 0.8 electron volt, and thereby it allowing to change their redox state very fast. In contrast, type 2 copper complexes usually show large reorganization energy, sometimes larger than 2 electron volt. Hence, rate of the electron transfer is so fast in blue copper proteins. Now, copper to copper 1 redox potentials are shown here. As you can see that these are the blue copper proteins isolated from different sources, and under physiological pH they varied their potential remarkably from 0.184 to 0.680. Please note that copper to copper 1 redox potential is only 0.16. So, it is high positive value in the blue copper proteins, and this is because copper 1 geometry is imposed on the oxidized site for the purpose of achieving a high redox potential. Now, next question is that how such a large potential variation is possible. So, this is basically this active site structure of blue copper protein. First, coordination pair also affects, and it can changes this redox potential along with the second pair interactions from the protein. Indeed, both are contributing towards such a large variation in redox potential value. It is interesting to see here, in the field of bioinorganic chemistry, the protein is an ultimate ligand capable of playing many roles in controlling reactivity. The blue copper sites clearly demonstrate a number of these roles in electron transfer function. Today, I have showcased how the rate of electron transfer is enhanced by many thousand folds in blue copper proteins as compared to the normal copper centers. The unique spectroscopic features of the blue copper proteins are the clear reflections of the role of protein chains in stabilizing the unusual geometric and electronic structure of blue copper site for its function. I also have illustrated here how such a unique geometry in both the oxidized and reduced states of copper accounts for the minimum reorganization energy and thereby enhanced rate of the electron transfer. The concept of the enstatic or rack induced state has also been discussed here. Thank you.